The next speaker is Ted Satterthwaite. He's actually the only person that I hadn't met before the conference, but as I said to him when we did meet, that several people in our group have a scientific crush on you for the reason that you've been so critical in a good way, methodologically, about the functional neuroimaging methods, results, and things like that. So we wanted you to be provocative. We wanted you to be critical, and we hope you will sort of challenge us as a field for how we do functional neuroimaging and draw the correct inferences from what we're doing. So thanks for coming.
Thanks so much for that really kind introduction, and I really appreciate you guys inviting me. This has been such a fun conference. I've learned so much. I've met so many fun, interesting people, and so it's just been a total pleasure. So I really appreciate you having me. Today I'll be talking about a little bit about some of the challenges and opportunities we have in translational studies for pain. I'm going to start with disclosures and caveats. I have nothing to disclose financially. My main disclosure is that I'm not a pain expert, but rather I mainly study neuroimaging, primarily in the context of brain development, large-scale studies of the developing brain and relationships to psychiatry. I got into pain through an awesome collaboration with the folks at Action, particularly Bob Dworkin, Rob Edwards, and others. And it's through them and their knowledge and enthusiasm and collaboration that I've gotten to work on this a little bit and ended up here. So thank you. So I'd like to start with a very simple question, which is what would make neuroimaging actually useful? And what's behind that question is the statement, it's not actually useful right now. And I think we need to start there. It's not actually useful right now. But what are our hopes? What are our dreams for neuroimaging? Well, we could start with the relatively simple one of localizing, where in the brain is this process pain happening? How about maybe slightly more ambitious, but that seems achievable, group differences, patients with chronic pain versus people without chronic pain? How are their brains different, functionally or structurally? But then we get into the interesting and clinically useful stuff. So I'm a psychiatrist, and whether I was seeing with someone with pain or someone with depression, it's not, these top two ones wouldn't actually be useful to me. But when we get to the lower ones, individual specific diagnosis, individual specific prognosis, telling a worried parent how worried about your kid you have to be, that starts to matter. And then even more exciting, biomarkers for the development of new therapeutics, reducing heterogeneity and finding new diagnoses, what's happened in oncology. We've gone from it's a tumor to it's a tumor from here with this cell profile and to respond to this therapy, and we're going to develop this therapeutic for it. So, and ultimately, that's really what I care about as a physician is treatment response, helping me choose a treatment that will help my patient. And so today, I'm not going to talk about any of the interesting stuff. I'm sorry. We're not there yet. We're just going to start with the top two. What brain regions respond to pain, and are there group differences in chronic pain? This is the outcome of 4 meta-analyses that we did in collaboration with the folks at Action, it was very illuminating. I'll take you through them. The organization is, I'll start with localization, fMRI responses to acute pain. Then we'll go into group differences and chronic pain, and then I'll talk about some strategies that we might think about moving forward. And I want to emphasize at the beginning, the strategies for moving forward are not specific for pain. I think they apply to translational studies in pain, but they're really kind of field-wide problems in any translational neuroimaging study, whether psychiatry, pain, something else. The first study was conducted by a really awesome data analyst who's now a bioengineering graduate student at Stanford, Anna Shu. She looked at a meta-analysis of fMRI responses to acute-induced pain. And here, this is a meta-analysis of studies of task fMRI. And I know many in the audience are not imaging geeks, so I'll try to give a very simple explanation. Let's say I'm lying in the scanner, and I'm just lying there looking at a crosshair. Nothing painful is being done to me. And then we're measuring brain response here, so that's at rest. And then I've got a transdermal patch on, and I get some sort of electrical stimulation, get a painful stimulus, measure brain response, then I will rest for a while, get pain. And really, we're trying to figure out, okay, what is the brain's response to induced acute pain? This is like the first level question. Again, not at all clinically useful, but at least helps us localize what brain circuits are involved with pain in a non-invasive way in awake humans without opening up their skull. So simple question, can we use fMRI to reliably localize brain responses to acute pain? So this was a pre-registered meta-analysis, and I'll talk about pre-registration a lot later, but basically what that means is we wrote everything down before we got started. And then like you can walk through the consort diagram here, but like screening 7,000 articles initially, then then that got down to a little under 5,000. And screened, then read 1,100 articles to determine the inclusion criteria. Then that was read by a second reviewer, so we had 2 people looking at 1,100 articles to ultimately include 200 articles in a quantitative synthesis and meta-analysis. This is a lot of work. So the key parts here, though, is that everything was written down in terms of the inclusion criteria from start to finish, and then the 2 reviewers. This kind of took a lot of play out of the system and ensures when we do pre-registration that we're not manually overfitting to what our biases are. We agreed upon it up front, and the data is going to fall where it falls. So among those articles, there's a lot of different study designs. This is a pretty broad inclusion criteria and includes men and women, all sorts of different stimulating pain in different ways and in different locations across the body. It's like thermal pain, non-thermal pain, mechanical pain, electrical pain, stimulated on the left side versus the right side, all male versus all female. So it allows us to look not just at pain more generally, but also some specificity analyses to see whether this circuit that responds to this pain could be, different under different contexts. And so the main result of the pre-registered meta-analysis was actually quite positive, robust activation of the pain network. And this is in some ways not surprising because this is not, if we go back to the inclusion criteria, remember, this is 200 studies that met a pretty rigorous inclusion criteria. This is not a new field, but at the same point, sometimes fields have been around for a while, they're pretty inconsistent. But what was remarkable here was it was quite consistent. And what we saw was robust activation in a really conserved set of brain regions, including secondary somatosensory cortex and anterior cingulate, thalamus, anterior and especially posterior insula. And really what was just a quite robust effect across all the studies. But remember, since we have all these different studies included, we can break it out by different types. And I'm not going to take you through the million supplementary figures in this paper. You should definitely look at the supplemental if you're interested. But at the same point, a couple illustrative ones, we could look at thermal versus non-thermal, electrical versus mechanical, males versus females. But the bottom line is, there weren't really big differences in what was activated under these different conditions or these different populations. You basically, if you look at electrical versus mechanical, it's the same brain regions that you saw in the overall analysis. It's just you have less power because there's less studies. So in conclusion, there's convergent bilateral pattern of pain-related brain activation. The regions are really similar across studies, really regardless of the stimulation technique or participant sex, and it really suggests a highly conserved core set of pain-related brain regions. Okay, so that was the easy one. That's almost a straw man of a study. We know there's certain parts of the brain that respond to pain. Voila. What about group differences? Chronic pain versus people without chronic pain. Okay, getting a little harder. Still not actually useful, but getting harder. So we did 2 of them. First is fMRI and then brain structure. The first was again led by the exceptionally capable Anna Hsu. And here, again, task fMRI responds to acute pain, line in the scanner, something painful is happening to you. But here, instead of saying what in the brain responds to pain, we're going to say what in the brain is the brain responses to pain different between people with chronic pain and people without chronic pain. Is that brain response different in the context of chronic pain? But remember, the stimulus is acute pain. So, there's lots of reasons you could think either way. This is going to go, okay, people with chronic pain are going to have a more acute response to induced pain. It seems plausible. So here, this paper came out, I guess, 2 years ago now. Same sort of design, fully pre-registered, 2 reviewers. Started with not quite as many records, around we got down to like the abstracts in terms of, we screened around 3,600, again, 1,100 full text articles, similar to that at that stage. But then instead of 200 articles, we eventually only included 29 that met the inclusion criteria because a lot of them didn't have the right group comparison that we could kind of meta-analyze across. Also, I want to point out that there actually were 47 studies that could have been included, but we only analyzed 29. The reason for this is one of the primary limitations of imaging meta-analyses. Like, I had not done a lot of imaging meta-analyses before this. I was kind of shocked that this is a known limitation in the field, which is for imaging meta-analyses, current methods do not allow to include studies with negative results. Seems like a problem. So right off the bat, you were incredibly biased towards finding something because we can only meta-analyze studies where something was found. There's not like we can't include a study with a null result like a typical meta-analysis because there's too many outcomes across the hundreds of thousands of locations in the brain. So this is a problem we acknowledged at the beginning, and there's 29 studies we can look across. And if we just look initially, okay, chronic pain versus no chronic pain, who activates more? And you see that the location and the direction across these studies varies really widely. And initially we saw there, like, I don't know, who knows? And when we do the main pre-registered analysis, however, we found there was no significant results. And this really kind of lines up with this map here. Like, you can kind of squint and maybe see something, but there's not like a clear, it's not like it's all red, more activation in chronic pain, or all blue, less activation in chronic pain. So there's no significant results in our primary pre-registered analysis. However, on the advice of collaborators and our very thoughtful anonymous reviewers, we conducted some unpre-registered post-hoc analyses, because if you look at the unthresholded results, and what I mean here is the way we could correct for multiple comparisons in imaging. So typically, if I were to run a clinical trial, I would have one primary outcome, and I would write it down, and I would run statistics on that primary outcome. In imaging, we typically look at the whole brain, and there's 200,000 locations, so we run 200,000 statistical tests. And that means we're highly likely to find false positives simply by running a lot of tests. Type 1 error, false positives, we can see something. So there's now, and I'll get to this later, field standard techniques for addressing all these multiple comparisons for looking across the whole brain. And that's what we found. When we use these field standard techniques, we found no significant differences. If we just look at the results without correcting this for those multiple comparisons, however, you do see something. You see that there's maybe chronic pain is a little higher in some pain, in like regions that make sense, like the insula, cingulate, in chronic pain than not. So based on that, we actually overlapped it with the results of the previous study, where we can see, okay, this is the pain network that activates to acute pain, and here's where the group difference is. Like, oh, okay. So we took one outcome, how much do you activate that pain network, and just looked at that one outcome between chronic pain and healthy people. Again, though, bold font, not pre-registered, not planned, post hoc, take with lots of salt. And what we found, though, is that in this pain network, in this post hoc analysis, patients with chronic pain do have a greater response to acute induced pain. So to conclude, for this study, we found no differences in our pre-registered outcome between chronic pain and healthy comparators. An unplanned post hoc approach did reveal significantly greater responses in the pain network in chronic pain. But we had a lot, I just want to say we had a lot of statistical power here. We're looking across 29 studies, and remember, right off the gate, there's a bias for only including positive results. So if there is a significant difference, it is subtle. It is really subtle. You need subtle statistical techniques to bring it out. It's probably not a big thing. So what about brain structure? We've been talking about fMRI the whole time. You can get a lot of different types of data out of the magnet. Every fMRI study has T1 images to look at brain structure because we have to line them up. So like, this is actually an even bigger field. Alina Henn, an awesome grad student working collaboratively with Simon Eckhoff's group in Germany, took this on, probably regretted it at various points. And here, Alina is asking, do patients with chronic pain have alterations in brain structure? So same question as the last study I showed you, but here we're looking at brain structure. And I'm saying this is a broad basket. There's a lot of different structural measures. The most common measures we looked at here were cortical volume and cortical thickness. So cortical thickness, you can see, is simply just the measurement in units. There's a unit, which is nice, unlike fMRI, millimeters between this peel surface in red and the white matter, gray matter surface in blue. And you can just measure that. You get a map of cortical thickness across the whole brain, and then you can compare patients with chronic pain and people without chronic pain. So just like previously, pre-registered, 2 reviewers, even bigger pool here though. We had 9,000 studies to start, 7,000 abstracts, 2,000 full text articles, initially 98 articles meeting inclusion criteria, 14 excluded because of negative results, again, bias, and then analyzed 84 of them. Huge statistical power, 5,000 participants across all the studies. If there was something, we should probably be able to see it with a sample of this size. And the main results were no significant differences. And this was surprising and disappointing. So again, based on discussion and feedback, we did some post hoc analyses that were not planned using more sensitive statistical methods. And we saw something, again, primarily in the pain network, like especially the insula. We saw some cingulate stuff where patients with chronic pain seem to have a little bit less thick cortex, a little bit less cortical volume. But again, the main analysis was not significant, and we had a lot of statistical power. So the conclusion of this study is the same as the last one, which is that to the extent that differences are present in terms of patients with chronic pain or in terms of brain structure, it's likely pretty subtle. That being said, there are some similarities across the past study as well, which is that in both of them, even though we didn't, according to our pre-registered measures, we didn't find blazing results, but there were these subtle results, again, in the pain network, suggesting that if I were to have a time machine and do it again, I would have, focused a little bit more on the regions that we know are involved in pain from prior research, probably had better statistical power, maybe would have done something. But in some ways, that's the whole point of pre-registration. It's because you don't have a time machine. All right, so given this, I'm gonna spend the remaining time, and hopefully leave plenty of time for questions and for you guys to argue with me, for strategies going forward. And I want to come back to where we started, is how can we make this actually useful? And again, freely acknowledge that we only covered the 2 first ones, which are kind of lame, and didn't get to any of the actual useful ones. And the real question, though, is when I had this, okay, we found significant results for the first localization, but group differences, which seems like it should be a pretty low-hanging fruit, the pre-registered analyses had no significant results. So even before we get to any of the cool stuff, I want to talk a little bit about what are some of the reasons why this happens and we don't find anything significant when we look across the literature. And this is not just about chronic pain. If you look at the recent meta-analyses in depression, same deal. This is a general phenomenon across many brain behavior studies or symptom studies with translational imaging. And so all the things I'm going to talk about next are broadly applicable to translational imaging, not just about pain, but there's some certain cases where they might be particularly relevant. There's like 50, but I only chose 8. You probably will tune out after 3. It's okay. So I can go through 8 things, 8 reasons 8 ways things can go sideways, and some strategies that we might be able to handle. And throughout, I'm going to try to highlight psychedelic research. I know there's a huge amount of excitement around psychedelics. And the 4th study we did with Action, led by Sophia Linguidi and Jake Vogel, we tried to do a meta-analysis of acute administrations of psychedelics, but it was too heterogeneous and we couldn't do a quantitative study. So we did a kind of standard scoping review. And I'm going to try to weave in some of the stuff from the psychedelic study into these 8 challenges. So the first, pre-registration, we talked about it. Very simple idea. Think about what you're going to do and write it down first. There's so many reasons why this makes sense, and there's reasons why clinical trials require it, because we're academics and we're all human, and we all have our own biases, we all have our own incentives, we're all up against, have limited time, and want to have a certain amount of productivity that's valued by our departments and field and collaborators and ourselves. And so there's just an incredibly strong incentive to get results. And imaging data in some ways is particularly bad for that because there's so many measures you can get out of it. There's so many ways you can slice and dice the data. So if you analyze the data long enough, you'll probably find something. So it's most important, like pre-registration, could be argued is like really, really important for imaging just like it is for clinical trials. This was really brought home by a landmark study from Russ Poldrack's group at Stanford. It was published in Nature around 3 years ago, where they gave an fMRI data set to like 20 to 30 teams, I forget the exact number, and then they sent it to everyone, and they agreed on the hypothesis, and then they said, okay, test this hypothesis, and then send me the resulting map and tell me whether it's significant and what the interpretation is. And there's so much analytic flexibility in the different ways you can analyze this data. The results were like hilariously discordant. Everything from blazing activation and the one conclusion to deactivation, the opposite conclusion to no results. And so this is the best argument ever for pre-registration is write it down, do what you said, because otherwise who knows what you can get. Second, large samples. So this was really brought home by this paper that came out last year and caused a lot of consternation on Twitter and in conversation by Scott Merrick and Brendan Turbo-Clemens, Damien Fair, and Nico Dosenbach, came out in Nature. And basically what the argument was is that brain behavior associations the effect sizes are almost uniformly small. And because it's a small effect size, due to sampling variability, in smaller samples, you can get any range of results. And you need large samples to get a reasonable estimate of effect size. Like all of the follow-up and like firestorm from people who don't want to give up small studies was directed at them. And they basically were like, we published this like chapter 1 from a statistics book in Nature, sorry. But this is well known from statisticians, and most statisticians would say, why is this in Nature? But it's applied to a brain behavior study. So here, just to walk you through it, sample size on the x-axis, a correlation. Here, this is the relationship between like default mode thickness, like the cortical thickness in the default mode network, and cognition. So nothing pain related, but you can imagine this being like a symptom score or something. And the real effect size in the full sample, and this was replicated in other larger samples, is tiny and negative. But as it takes you hundreds and hundreds of subjects to converge to that in most of your subsamples, when you have less than a couple hundred subjects, you can get anything from a positive relationship to a negative relationship and anything in between. It's the problem of sampling variability with small effect sizes. I want to caution that this doesn't apply to all imaging studies. Certain designs, like within subject designs, where you have someone on a drug then off a drug, that's much more powerful. These results don't clearly apply. So it's not like all doom and gloom, but there is a lot of doom and gloom. So in terms of psychedelics, despite saying that you need large effect sizes, you look in the literature and these are small studies. And that's not a criticism. Those are really, really hard studies to do. I would never attempt it, so bravo, but they're really small. Next, false positives. We were talking about this earlier with type 1 error. So clinical trial, you look at one outcome. Imaging, I'm looking at 200,000 outcomes. Run 200,000 regressions, you're going to find something just by false positives alone, chance alone. So over time, and I showed you this in the uncorrected results, which didn't survive multiple correction comparison. And over time, this has like kind of been one of the worst things in imaging, but the field has largely gotten through this crisis. This crisis peaked in like 2015, 16, and has like kind of been relatively resolved from there. Previously to this paper by Anders Eklund, Tom Nichols, and others that came out in PNAS and was covered in the New York Times, like people were doing all sorts of different methods for correcting for all these comparisons across the brain. But now the field has really converged. There's good statistics to back it up, field standards. When you don't follow those field standards, there's this hilarious paper where people looked at activation in the brainstem of a dead salmon. You can definitely find stuff. But now there's field standards, and we know how to correct for multiple comparisons. It's not actually that challenging anymore, but it's like implementation science. It's like it hasn't had widespread like universal adoption in the community. So, for example, in the psychedelics review we did, we looked through, we basically graded the studies on whether they met kind of current field standard methods for type 1 error correction. And in some ways, it's totally not fair because some of these studies were done before that crisis was resolved. So short of a crystal ball, there's no way the investigators who conducted those studies could have seen the future. Again, no blame, just trying to see what was done. And around 60% of them did use current field standard methods, but that means 40% didn't. So this is a way how kind of inconsistent and unreplicable results get into the literature, and why you might run a meta-analysis with a ton of studies and find nothing because it was type 1 error to start with. All right, we're gonna move on to another bad one, motion artifact. This one's very close to my heart. I lost years of my life to this one. So most of my lab does developmental imaging, so we study kids, and I started working on it right when I had twins, and you don't have to do a large-scale developmental imaging study to recognize that kids don't sit still. You can just come to my house for dinner, and so and then you're asking them to lie motionless in a scary tube for an hour. So this was a very early plot that within an early study where you can see this huge relationship between motion, in-scanner motion on the y-axis and age on the x-axis. And you can see that there's this very strong nonlinear relationship that kids just move less and less as they get older, or they move more and more as they're younger. This is not just a concern for developmental imagers, though. Subsequent work, including this awesome study by Josh Siegel, showed that in-scanner motion is basically related to almost everything we care about. So whether it's BMI or cognitive status or symptoms, DSM diagnoses, smoking, high blood pressure, it's related to motion. And I don't know if anyone specifically looked at this in pain. I'm not aware of it, but it would not be a far-fetched guess to imagine that people in pain have trouble sitting still in an uncomfortable magnet environment. But okay, they move more, but who cares? The problem is that in-scanner motion for fMRI has an order of magnitude or more effect size than even the strongest biological variables of interest. And so to the degree that your subject level variable, your independent variable is related to motion and you don't control for motion, you aren't doing that study. You are just studying motion artifact. It is gonna be a very, very severe confound. We published this in 2012 synchronously with groups from Harvard and WashU, and it's just a massive effect on the data. When you see results this strong, you know it's technical artifact, not biology. So psychedelics, how are they doing? It's kind of like half and half. We went through and rated studies how much are they adhering to field standard guidelines, and we rated it from 0, don't even acknowledge it's a problem, to 1, acknowledge it's a problem and try to do something, to 2, follow field standard guidelines. There's a lot of heterogeneity and plenty of room for improvement. Some studies are really doing a good job, others not even discussing it. And again, just like in pain, if you give someone psilocybin and then have them be in a magnet, they might move more or less. I don't know. Probably worth reporting. One optimistic thing is that if you look over time, the methodological kind of adherence to current field standards has improved over time. Some plateau right when things are getting more popular, but this would be the case for not just psychedelics. This would be the case for depression, schizophrenia, whatever. All right. So given this, our work on motion artifact led us to get interested in pre-processing and methods to reduce the impact of motion artifact on our data, which in turn, so we started developing software and people started using it. And this led to concern about reproducibility. And to back up for a second, I'm an imager. I have a lot of friends who are basic neuroscientists. If I were to get most basic neuroscientists in a private conversation, especially after I've bought them a couple drinks or something like that, and ask them, so what do you really think about neuroimaging? Most of them would say, well, I don't believe that. And because we do so much to our data, there's so many pre-processing steps, there's so many things, there's so much data manipulation. And so I'm a very firm believer that sunlight is the best disinfectant. So in developing the software, we've put a ton of effort, not just into like reducing artifact, but making it all transparent. Like instead of the black box, the glass box. And so over time, this is all work led by a team of people led by Matt Cieslak, who's the director of neuroinformatics in the lab. And so over time, we've developed post-processing methods for fMRI, XCPD, processing and reconstruction methods for diffusion imaging, QSI prep, and methods for processing and quantifying cerebral blood flow with arterial spin-labeled MRI, ASL prep. And so these methods are like try to show you every step what was done to the data. They automatically configure to apply the appropriate methods based on the metadata in the images themselves, so there's not as much analytic flexibility for the user. They provide boilerplate processing reports. You can just paste it into your methods section. And they provide a lot of QA things, so you can covary for any quality measure in your analysis. Furthermore, they're containerized, and that's a software term that means that all the dependent software is wrapped up in a container so you can deploy it, whether it's on your laptop or the Michigan computing cluster, the Penn computing cluster, whatever. And so a lot of people have used it. The problem is that this is just processing, and things happen before processing. When we do an imaging study, there's usually a coordinator, very hardworking, very well-meaning, who makes decisions about the data before it's actually analyzed. Fixing this piece of metadata that was missing from the scanner for inexplicable reasons, removing this participant because they had like there's something weird that happened that day. But usually those sorts of curation decisions are totally manual, and they're written down in a lab notebook and blue pen in a drawer somewhere. And so we developed software called Qubits to reproducibly curate large-scale imaging data sets so you know what's in there, and any curation decision can be totally tracked. The way it's tracked was with a software called DataLad. DataLad is a relatively recent software tool that tracks data just like Git tracks code. Code, typically software engineers, they track code using software called Git. So every little change to your code is tracked. You know who did what when. It can be rolled up, rolled back. Many labs use it. All the software companies use it. This is a method that allows you to do the same thing with data. So any change to your image, byte for byte, can be tracked across the entire workflow, know what was done when, complete audit trail, roll it back, roll it forward, totally reproducible. And so this is the problem with DataLad is it's really hard to use. It took like a senior postdoc 2 years to really get comfortable with it. So we made another software package to wrap it up and try to make it easier for people. It probably still is not as easy as it should be, but we're working on it. All right, next, switching to biology away from methods. Why are we not seeing convergence of these meta-analyses? Maybe because the biology itself varies across humans. And one of the most, I think, important developments in network science was led by a team at WashU that found that canonical functional networks vary a lot across individuals. So typically when we analyze networks, like for example, here's the default mode network, here in this rose color, same in red in this atlas, we basically line up the structural image, and then we assume that by lining up the structural image, we've also lined up the functional brain networks. Seems reasonable. The functional brain network should be on the same part of the cortex in everyone. However, Tim Laumann, Nico Dosenbach, Steve Peterson, and Damien Fair showed that this is just demonstrably false. And initially into this N of 1 study in 2015, and then a larger study, like N of 10 study in 2017, now replicated by many groups across many methods, different data sets, shows that the location of functional networks varies a lot across humans. We all have our own individual specific functional neuroanatomy, and that it varies a lot, and it varies often even more in the parts of the brain that we care about, or at least I care about, like higher order association cortex. The visual system doesn't move around as much, but like the default mode network moves around a lot. And so like this is basically saying that simply lining up brain structure does not line up brain function. And that's a problem, but it's also an opportunity, because maybe one reason the meta-analyses aren't landing is because the functional circuit is in slightly different places in different people, so you're blurring it enough that you can't see it. you can actually look at this, what's called functional topography, the location of these functional networks across people, and relate these things to brain and behavior, related to behavior. So Zaishu Kui, an awesome postdoc in the group, related the location of these individuals' networks and their relative size to each other to something we care about, executive function, a common broad cognitive domain. And he showed that kids with bigger executive networks are better at executive tasks. Furthermore, if you look at the overall multivariate representation of an individual's specific functional neuroanatomy, you can predict their executive function in unseen data with pretty good correlations, over 0.4 in both halves of the data. So personalized functional networks are one potential way, not purely method, but actually biology that we might be able to move forward and get more consistent results, especially if you're interested in higher order association networks. However, probably the single biggest one of these 8, if you want replicable results, number 7 is replicate it yourself. And this is how it's worked in genetics for a long time. You can't get a high impact paper without discovering and then replication. You need both samples. And imaging is pretty reliably, a ways behind genetics, but we're starting to catch up now. And fundamentally, I like, the students in the lab make fun of me because graduate students and postdocs are often more excited about a new shiny result, but I don't want new results. I want a replication of an old result so we can actually believe it. It's not always that popular stance, but the one result I'm super excited about was that result I showed you previously. Was this one, Zaishu relating personalized functional networks to executive function and unseen data using our local data set PNC, around 900 kids in that one that we analyzed. Here, this is the ABCD sample, same results, same method, same effect size, completely different data, generalized beautifully. Much more likely to actually be true. This is work from an amazing postdoc, Ariel Keller. And just to end, this stuff is hard, like pre-processing data, QA of data, it's hard and unsexy work. No one wants to do it, honestly. So the project I'm most excited about going forward is creating data resources that makes it easy for other people to do good work and quickly discover and replicate their findings. And so this is a project I lead together with Mike Millam at the Child Mind Institute called the Reproducible Brain Charts Consortium, where we'll include many of the largest cross-sectional studies of brain development, do really detailed quality assurance, all pre-processing where every single thing is tracked. We put every piece of raw data, code, process data, QA on the internet, really light license so people can just download it quickly and run. The goal is to make the entire workflow reproducible, because in the end, if you want people to believe you, the best way is to show them everything. And so my hope is that not just sharing code, but sharing data allows us to happen. Fundamentally, I can give you a link to my code for my paper, but you can't replicate my findings unless you have the input data. So hopefully, with these sort of data resources, you can really go from nose to tail and believe it for yourself. The first project that we've used this data set for is a work by Audrey Lowells. And here, she used this data resource to show that functional brain development develops along a sensory-motor association axis reproducibly in 4 data sets, first discovered in the PNC, pre-registered, and then replicated in 3 independent data sets. And by the end of it, we're all pretty convinced this one's real. So I'll stop there. Want to leave plenty of time for questions, but mainly I hope I haven't depressed you too much. I was told to stir the pot, and I tried to, even though maybe it's not my natural inclination. So thank you guys for having me. This has been a really fun conference.
Thanks so much, Ted. The amount of truth per unit time was just off the charts. So on the pain front, I know you're not going to be here this afternoon, but my within-person analysis of fluctuations in person's chronic pain with brain networks maps really nicely onto your meta-analysis of healthy individuals with provoked pain. And so I think that probably when you don't see differences between chronic pain patients provoked by pain and healthy controls provoked by pain, it's probably just the heterogeneity of the provocation and how well it aligns with their pain experience that may account for that lack of difference.
That makes a lot of sense.
Is the Andrew and I published last year a study of cortical surface area, and that didn't vary by patients versus controls. It only varied by essentially those with nosoplastic pain versus those without. So I think a lot of the heterogeneity can get washed out of those meta-analyses too. So just wanted to see if you had any thoughts on that. But really beautiful work.
Thank you for the really kind words. And that is, I think both of your points are completely valid, well taken, and true. And the analogy to a field I work more in, like psychiatry, like heterogeneity is everything in depression. And like, it's probably not, depression is not like one thing, it's probably many different things. And unless you take like account of the patient-specific clinical context, patient-specific anatomy, it's just very hard to find things. So I think your points are extremely well taken, Jason. Thank you.
Andrew?
Right. So first of all, that was a fantastic talk, and I'm only like a little sad. You hit the right balance. So I have a question about the merit paper that was in Nature, because this obviously comes up a lot, right? It suggests so much that we
You could go back to the slide that Andrew's referring to.
And there's just one thing that's always bothered me about that, and I know it's not your paper, but maybe you have thoughts on this. And, essentially what they did, right, they pulled out subsamples, ran the analyses, and acted as if that was its own little study, and what if that was little studies. But when you look at the supplemental material, you look at some of those things, one of the things you can see clearly is that the range of cognitive performance is really truncated in a lot of those samples, as you would expect. And if I was going to study depression, I'd want people who are not at all depressed and people who are really depressed and everything in between. So when you pull in a sample and everybody is either not cognitively impaired or a lot of them are, I wouldn't look in that sample for a brain signal because I don't have an appropriate range in the predictor.
You would have designed a better study.
Well, basically, let's not interpret this.
Totally.
Because we don't have a range of depression.
Yeah.
We're going to look at anything in the brain. I sort of felt like, as a sensitivity analysis, they should look at a range of the prediction to see, is it truly sample size, or is it just that as you get up to 100, 150, 200, 1,000 people, then you have a beautiful representative range of predictor. And I've never seen that addressed.
Yeah, they didn't. I agree. They did not address that in the paper. I can't pull up which group did it, but there has been a more recent package that allows you to stratify subsamples more evenly to make sure in cross-validation that you have that nice range. Because you're right, the psychometric properties of trying to run a correlation on something where there's no range of the data, impossible. Can't do it. So I think I think you're right. I don't know how much that would affect their results. I bet Scott or Nico would do that if you contacted them, or at least argue with you on Twitter. But there are tools available to do that now easily for imaging analyses, because you're 100% right. That's like an obvious problem that is unavoidable. If you don't have the range in your data, you can't make a good prediction.
Owen?
Amazing content. Just kind of, what are your thoughts on the balance between sample size in terms of number of participants and duration of scan, visual resting state connectivity, like a bit less time for a small number, but really long scan times? What's your thoughts?
Yeah, great. Absolutely. So Owen's question is length of, I realize I should be repeating this to folks for the people in the other room. Owen's question is about N in terms of the number of samples versus kind of quality of the measurement, the duration of a scan for like fMRI. This is a big trade-off, especially because people usually have multimodal imaging studies, and they want to pack in other things, and you'll have like a friend who really wants the diffusion data and someone who wants an iron image or something like that. And so you can't just get like 50 minutes of rest, and also the participants will really hate you then. And so I think there's a couple things. First, more data, always better. Second, depends on what you're trying to do with it. So the Midnight Scan Club paper, they show how much data you need for these individual specific networks, and it doesn't plateau, especially at some noisier networks, until a lot of data. That is a pretty unforgiving method, though. There are other methods that have kind of stronger priors that converge more quickly and are a little less data hungry, but it really depends. Like some of the like amplitude measures, way less data hungry. So you can get, I think just knowing not all fMRI measures are the same, knowing what the test-retest properties of your measure is essential. You can't just go by like in general. Second thing I would say is, so more data is better. Second thing is like, it doesn't always have to be rest though. There's like really good evidence that getting measures of networks from task data, whether you regress out the task or not, is super, super informative and helps like your statistical power a ton when for inference. I think the best paper that I know of from that one is by Max Elliott and Ahmad Hariri. Basically, they call it generalized functional connectivity, where you construct the network with all the fMRI you got available. And so all of a sudden, instead of your 6 minutes, you might have 20 or 30, and then you're really, really doing better. But in the end, you want a bigger budget. You want more time with the scanner. You want a bigger N, and you kind of often have to do the best you can.
Great. Thanks so much, Ted.